The goal of these procedures is to evaluate sperm membrane's integrity using specific fluorescent probes combined with fluorescence microscopy or flow cytometry analysis.
These techniques might help answering key questions in the field of reproduction, such as prediction of fertilization potential and quality of sperm sample. The main advantage of these techniques is that they enable precise evaluation of multiple sperm membranes, which are known to associate with sperm fertilization potential. The implications of these techniques extend toward diagnosis of ejaculate quality, as it enables more accurate evaluation of sperm plasma and acrosome membrane integrity, as well as mitochondrial membrane potential.
To begin this procedure, obtain approximately 1 to 6 milliliters of bull semen in a 15 milliliter tube at room temperature. Add 6 milliliters of NKM buffer, pre-warmed to 37 degrees Celsius, to each 1 milliliter of semen. Centrifuge at 600 times G for 8 minutes. Repeat this centrifugation once or twice until the supernatant is clear. After this, immediately discard most of the supernatant, leaving approximately 1 centimeter of supernatant above the pellet. Carefully lean the tubes at a 30-degree angle in the incubator and wait 20 to 30 minutes to allow the spermatozoa to swim up. Using a micropipette, carefully remove the remaining 1 milliliter of supernatant, which now contains the motile spermatozoa, and transfer it to a fresh 1.5 milliliter tube. Keep the sperm at 37 degrees Celsius.